Thank you for the invitation, Nikki. Thank you, Dr. Irwin, Dr. Shrestha, for the invitation. It's a huge pleasure to be here with all of you. I'm going to talk a little bit about heart failure. And I understand if none of these words on the slide make any sense, it's not about that. This talk is bigger than that, and we'll talk a little bit more about that. When I talk about heart failure, it's not born in the cath lab or in the echo lab, some cardiologist looking at some grainy images and saying the squeeze is not good and it's not doing the job as well as it should. It's sculpted by genes. It's incubated in the neighborhoods, and it leaves an imprint on the sarcomeres. And I'm here to talk more about how I explored that. A straw poll. How many of you are familiar with the term epistemology? Not epidemiology, epistemology. Oh, there's one person. Good. You don't have to worry about it. It's just fancy jargon that we academics try to create so that we can't make the common man understand what we're doing. It's basically the theory of knowledge, how we know, what we don't know, what is true, what is likely to be true. We see problems, we present conjectures. It's a falsifiable conjecture. We got to prove it whether or not it works. And we learn from it, whether we accept that conjecture and move on, or we stick to whatever explanation that is best available in that very moment. Nikki mentioned the beginning of infinity, and David Deutsch is a father of quantum computing, and he's one of my favorite scientific philosophers, and he says, the search for hard to vary explanations is the origin of all progress. We don't arrive at truth in one stroke. We circle, we criticize, and we refine. Every new law of physics, every medical breakthrough was always outlandish. And I'm here to inspire you to recognize that scientific revolution has showed us that progress is possible if we're willing to learn, to question, to fail. And this talk here's how I approached it, how I made all those mistakes and pursued constant error correction. The focus is not what statistical model I used, what paper I published, what fancy omics technology I used. Sorry, Nikki. It's about how I approach this. No financial disclosures, unfortunately. Nobody wants to fund the work I have done yet. But upside, intellectual freedom. A little bit about me. My parents are both internally displaced Kashmiri refugees. I was born in the foothills of Himalayas in Dehradun. That's where I did all my schooling. I then went to Dharamshala, where I did my medical school, which is again right in the middle of Himalayas. And then I came to the lower Appalachians here in Birmingham. I came here first as a visiting medical student. I then ended up doing a research fellowship, and eventually now doing cardiology fellowship in clinical cardiology. Growing up, geography was my favorite subject, and you'll see this theme come up again and again. My journey has taught me through all these places place is not where you live. Oftentimes it is what you become. And I'm going to try and prove that conjecture through my work. My story and my family's story is about finding meaning in noise. When you're surrounded by noise, you got to find that hard to vary explanation of what's happening around you. So let's start with why heart failure, and more importantly, why now? I love this wonderful graph from King, recently published in JAHA. On the left-hand side, if you see the panel, 91% of deaths from cardiovascular causes were because of acute myocardial infarction or chronic ischemic disease. Back in 1970, when we actually started tracking this more robustly. It's fallen down tremendously, and it's now 54%. 89% to an 80% fall if you look at each of those categories. And there's a lot more of this other heart disease that has happened. And if you understand, heart failure is that end product of all these illnesses. Either you die from a heart attack immediately, or you survive through it and the heart keeps going through that remodeling changes, and the end stage is developing heart failure. On the left-hand side, you see there's a 146% increase in coded diagnosis of heart failure mortality. And there's more, 106 of hypertensive heart disease, which we call in clinical terms HFpEF. That's all cardiomyopathy again. And there's an overall 140% increase over the last 30 years. We've moved away from ischemic disease and more towards this end-stage cardiomyopathy now. Heart failure accounts for nearly half of cardiovascular deaths, and probably even more if you account for all the hypertensive heart disease, the arrhythmias. Here's the stat, which really boggles my mind, is 1 in 4 of you in this room is going to develop heart failure at some point in your lifetime. Let that sink in. That means it's a problem we haven't been able to mitigate, despite all the therapeutic advances in the last 2-3 decades. 7 million Americans still live with heart failure, and that number is going to keep going up, 11.4 by 2050. And it leads to an estimated annual expenditure of 142 billion. You're not going to be able to tariff your way out of that. So heart failure is where upstream risk and downstream inequity converge. So maybe our explanations are not complete. And that's why I started to look at the exposome. So those of us not familiar with the term exposome is essentially everything that you're exposed to from the moment you're born till thereon. And you see there's the external exposome, which is where you live, the green spaces, the air pollution, the urban environment, the noise, the traffic. And then there's a lot of personal exposome, things that you did to your body, physical activity, your diet, smoking, your social interactions. And all of those lead to some biological responses that may be in form of gene expressions or epigenetic changes or inflammation. And all of them eventually cluster into this final product of heart failure. The image on the right is a cardiac MRI sequence of end-stage heart failure. I just find it extremely cool that you can visualize it and see what happens. Those walls that you're seeing, they're supposed to obliterate completely, and that's not what's happening. Let's dig a little bit deeper into heart failure. So when I came here, I was very fond of geography, as I said. So I wanted to find out where more people have heart failure. And you see, this is one of those maps where you're on a spectrum from 800 per 100,000 to 1,200 per 100,000. There's a lot of clustering in the Midwest to Northeast states where more people have prevalent heart failure. We are not that far behind in the South. But what's interesting is how many people die of heart failure. That is not there. It's a lot more people dying from heart failure down here in the Southeast, a lot more in all of the Southeast states. And I looked at it a little bit further. I wanted to cluster them into groups and see how that looks like. And in this work, we kind of clustered them into quartiles of cardiovascular mortality, similar work for heart failure mortality. And you see this, the Southeast states is where the highest level of cardiovascular mortality and heart failure mortality is. All right. I'm going to go through this slide in detail. No, I'm kidding. That's the busiest slide I've ever created, or probably you'll ever see. That was one of the figures in the papers that I wrote. But the big picture message was all the cardiovascular risk factors of diabetes, hypertension, hypercholesterolemia, obesity, smoking, physical inactivity, presence of ischemic heart disease and stroke was highest in the states of West Virginia, Alabama, Mississippi, Louisiana, Georgia, Arkansas, Kentucky, Tennessee, North and South Carolina, and Virginia. Those states seem oddly familiar to a lot of people here in the School of Epidemiology. We describe that as the stroke belt. And you'll see that a little bit more, how these cardiovascular risk factors kind of pool together with geography. I looked at the prevalence of poor cardiovascular health and how it's gross ecological association with cardiovascular mortality. Similar results with heart failure too. And you see that blue cluster of the South states. The poor prevalence of cardiovascular health was quantified using AHA Life Simple 7. But the big picture message was, we have poor cardiovascular risk factors, poor health behaviors, and higher heart failure mortality in the Southeast. And I was really intrigued by, is this still true? Is it that stroke belt still applies to heart failure too? And I looked at that. And the dark blue line is the stroke belt population. We've had a substantial improvement in the people dying from heart failure. And it's kind of reached an inflection point since 2011. But we are still 20 to 30% higher mortality in the stroke belt from heart failure compared to rest of the country. And that's the absolute difference, ranging from 12 to 16 deaths per 100,000. I was more intrigued by who is dying from heart failure in the Southeast. And the interesting part was, you see, they're clustered across these age groups, and it's not really the less than 24, it's the demographic dividend of the Southeast. 25 to 44, 45 to 64, the ones who are supposed to contribute to the economy of the Southeast. And it's growing up, increasing. The graph in the bottom left, that's the one which was the most appalling for me. Age more than 65 is still there, and that has essentially not changed. That rate is still the same. So on a gross scale at state level, we recognize there's higher heart failure mortality. And what I was really intrigued by at that time was, is this occurring even at the neighborhood level? I was, one of my colleagues at that time during my research fellowship was Gargia Mala. A lot of you may know her. She's worked in the Regards projects a lot, and she's now a cardiology fellow at Arizona, and she was interested in looking at neighborhood social determinants of health and how that is associated with incident heart failure. And sequential modeling, including demographics, health behaviors, risk factors, your consistently higher neighborhood social determinants of health clustering was associated with a higher incidence of heart failure. So there was something there that we are missing, we're not recognizing. I looked at it as well at the same time. Most people admitted with heart failure develop acute kidney injury, and that's the next most important organ that gets impacted. And we saw that people who were from poor neighborhoods, from low-income areas, with more rurality, had higher odds of developing acute kidney injury, and also had lesser chance of recovering from that because they are not able to seek the care after that first hospitalization. And it was true for just heart failure hospitalization and heart failure associated AKI as well. So geography is not just a proxy. It is one of those patterns and hard to vary explanations that keep coming again and again. And when you find something that keeps coming in again, you got to dig deeper. We understand geography predicts mortality, poverty, segregation, redlining. These exposures leave scars on the myocardium and leads to higher incidence of heart failure and worse outcomes among those who have heart failure. But geography is that one hard to vary explanation, which is also measurable, persistent, and consequential. And I kept thinking about this. This cannot be biology. This does not make sense. It's just access issues. Maybe there is more to it. Maybe context modifies biology. And at that time, I was working, my research fellowship was in a lab which was focused in natriuretic peptides. So I was focused on these natriuretic peptide handicap groups. For those of us who don't know natriuretic peptides, these are hormones produced by our heart. A heart can be an endocrine organ that helps in regulating your blood pressure, your cardiometabolic health, how you manage salt, hypertension, your kidney. And we know that people with West African ancestry have around 20 to 30% lower levels of this good hormone, and people who are obese have 20-30% lower. So those were the handicaps that I was focused on. So yeah, these are also the risk factors I understand for higher heart failure mortality. So I thought maybe those racial differences in natriuretic peptides, that maybe that's it. That's the reason why we have this higher burden in the Southeast. And we looked at people who were very cleanly phenotyped with heart failure. This is one of the trials, guided trial. Don't focus on the results. The big picture message out of this was across different levels of hormones, of whatever from less than 100,000 to more than 3,500, non-Hispanic black individuals had higher rates of cardiovascular mortality and all adverse heart failure outcomes. And this is after extensively accounting for demographics and clinical risk factors. This is a trial population, extremely well phenotyped. These people are followed every few months, and still we are seeing this pattern. So I was intrigued. So I wanted to look at other natriuretic peptide handicaps, and obesity is one of those NP handicaps. The graph on the left is the association of adverse cardiovascular events and with BMI. At that time, we used to believe in obesity paradox, that a higher BMI is kind of protective. We didn't see that, and now the literature has completely changed, and now we give Ozempic for conditions like HFpEF. Again, we saw the similar message of no matter what level of BNP you achieve, you were still having higher, worse outcomes among this natriuretic peptide handicap group. So there was more to it. Just taking us back in time, around 3-4 years ago, there was a huge push for circadian biology, and that's how sleep got integrated into the AHA Life Essential 8. For just to make sure everyone is on the same page, your blood pressure has a diurnal rhythm. It goes up and down. And I was wondering, maybe that has something to do with eventually causing heart failure. Your blood pressure dips a little bit at night, and when it does not dip, or it dips excessively, those are considered high-risk phenotypes. And I don't know if you've learned about the SPRINT trial yet, but it was this landmark study where we understand now why we have to treat to a blood pressure goal of 120. And even in this trial, where people were getting treated adequately for their clinic blood pressure, a high-risk nocturnal blood pressure type was associated with worse clinical outcomes. I was, at that same time, I was doing a physiological clinical trial, and I was like, let's go for it and look that this has to be the explanation for heart failure. I proved first in atrial tissue and left ventricular tissue that you see the NPPA and NPPB, those are the genes for ANP and BNP, the natriuretic peptides. Their expression is lower in obesity. And then I did a detailed physiological study where we recruited 40 individuals, 20 lean, 20 obese, did a very detailed protocol. They have stayed in the hospital. We got multiple blood draws. But we saw on the top left, the picture A over there, the dotted line is the obese population. There's a less dipping of blood pressure at night. And that is what we anticipated was there's these obese individuals with clustering of risk factors, very young, healthy individuals, so they didn't have any cardiovascular conditions, already show a phenotype which was worrisome. And this kind of overlapped with the rhythms of natriuretic peptides, BNP, AMP, and was exact opposite of renin and aldosterone. There was some circadian dissonance between the two, and that's another area of exposures. Maybe we fix the circadian biology. All of you must be bored of me saying natriuretic peptides. And that's what I was thinking. It was like that one thing that I kept looking at constantly. I was kind of blindsided. The Kentucky Derby horses, just had the blinds on, and I just kept going forward, like this has to be it. But if you're a hammer, everything looks like a nail, isn't it? So it could not have been. Biology cannot be just one thing over and over again. It has to be more. There has to be context. There's things that shape biology. And heart failure science needs us to compound our knowledge. So what about genes? I thought that you can't get rid of that. You're born with it. Maybe it's genetic predisposition that leads to all of it. Nikki there is smiling because I'm talking about genetics now. But you can't negate that. So I thought, let's look at it with this perspective. And my big conjecture was maybe genes load the gun and the context pulls the trigger. And we started with this cluster of risk factors that we kind of outlined: metabolic syndrome, incident diabetes, and social determinants of health. We understood that we know that non-Hispanic black individuals, due to a multitude of reasons, have higher incidence of diabetes, one of the most common risk factors for heart failure. So again, back in SPRINT trial, we went back and we looked at it a little bit more, and we showed that despite adjustment, that was still true. In this well-phenotyped, rigorously followed up cohort, they still had 40% higher risk of developing diabetes. Interestingly, they had a better HDL profile, better triglyceride profile. So it didn't add up. So I said, let's dig it a little bit further. Maybe it's if you look at the whole scope of the ancestry and just take a snapshot picture, does that make sense? And the less European ancestry you had a higher incidence of diabetes after extensive adjustment. And I said, that does not make sense completely. Maybe it's the clustering of social determinants of health in these populations. I looked at that. And there was like an entire spread of it. It wasn't one thing for everything. So maybe it wasn't one thing. Maybe it is both. Maybe it's more than just a simple answer. We understand that ancestry encodes metabolic fingerprints. So maybe it's genes and social determinants of health together. So I said, well, this is a lot. This is a lot to do. There's so many ancestry, so many genes that I have to go after. So I wanted to narrow things down, and I went to a single gene disorder. This publication in JAMA, we looked at cardiac amyloidosis. We have a better recognition of this phenotype of heart failure now. It's a condition where your body misfolds a protein called amyloid and deposits it in all the tissues, including your heart. It can either be because of advanced aging, or it can be due to inherited mutations in this gene, the TTR transthyretin gene. And there's this one variant which is prevalent in individuals of African ancestry, and this is around 3%. It's been reported in smaller studies that had some association with heart failure, and we wanted to answer that question and answer it for once and for all. So we looked at regards, we took carriers of this variant and non-carriers, and after adjusting extensively for demographics, prevalent and incident CHD, cardiac risk factors, health behaviors, and AFib, even SDOH measures, there was a consistent signal of 2.5-fold higher risk of incident heart failure and 40% higher risk of all-cause mortality. This work was one of the most consequential ones that I had done at that time. It led to reclassification of the TTR gene in the ACMG secondary findings list. That means if anyone gets a clinical genetic test, they will screen for the TTR mutation. This was great. Like we said, I was able to hone into one heart failure phenotype, and I dug in deep and I tried to answer it better. But I kept thinking, that's just one phenotype. There's so many more. I gotta keep doing this one after the other. I'm gonna exhaust myself. So maybe I should go after a risk factor. Let's find the one that is the most important one. And we understand that hypertension is the leading attributable risk factor for heart failure. So I said, let me look at genomics from the heart failure, from hypertension perspective, and try to go after the heart failure from there. I started simple because I didn't have a lot of omics skills like Nikki does. So I started with something simple like a candidate gene approach. Again, my blind sites were still on, so I was focused again on natriuretic peptides. And there's a genetic variant in corin gene which processes natriuretic peptides, and if there is a variant, you don't process it very well enough, and this variant is clustered among individuals of West African ancestry. Around 13% of them carry this. And at that time, there was a notion from smaller studies that maybe that's the reason why people from Southeast, where there's a higher burden of West African ancestry individuals, maybe that's why they have hypertension and then eventually heart failure. We looked at it in regards and in Jackson Heart study. And we verified it is 12% of African individuals had this variant. It wasn't associated with any of the hypertension traits, neither with incident hypertension or with natriuretic peptide levels or with gene expression in heart tissue. So you see, even in this case, I was constantly trying to prove it on multiple levels, whether my conjecture is true or not. And it has to stick and make sense. I know a lot of you in the room are thinking, but hypertension is not monogenic, it is a polygenic thing. And that's where I worked in the next scheme of things, the polygenic engine of heart failure being hypertension. Again, I didn't have a lot of skills, so I did with whatever I could and whatever resources I could gather, and one of those was a CORD trial. This was a trial back in 2000s where we recruited people with diabetes, so a risk factor for heart failure already. And we tried to prove whether or not the blood pressure cutoffs work for them. But this trial provided us with an opportunity to look at their genetic data, a very well phenotyped, well followed up cohort. And we took a small polygenic risk score of around 1,000 SNPs. So SNPs being, one mutation, small mutation at various spots across your genome, which are associated with a particular trait. And this is a polygenic risk score essentially becomes a genetic report of what is your likelihood of having a particular trait. I'm trying to keep this very simple because that's how I try to envision all of this. It gets too complicated too quickly. Our main outcome was ASCVD, so people dying from MI, stroke, CVD. And we looked at how different were those people who had the primary outcome and those who did not. There was a very subtle difference. But even that subtle difference after adjustment was still 12% higher risk of developing these primary outcomes. And that made me more intrigued. It's like, hey, I think this is now first time I'm starting to see a signal here. Maybe that polygenic engine works. So I was hoping at that time, hey, why just 1,000 mutations? Why can't I just capture the whole genome? And so I went after a genome-wide polygenic risk score. Created it and validated it in the top mid cohorts, ARIC, MESA, Cardia, CHS, Framingham, validated it again in all of us. And of course, the polygenic risk score was associated with hypertension, but incidentally, it was also associated very cleanly with incident heart failure, incident ASCVD. So there was the evidence that I was looking for, that polygenic engine of heart failure. And you see the effect estimates pretty similar to what we saw in the other study. But I took this a level further. I said, but you are born with bad genes. What about if you control your other risk factors, you control your health behaviors? This is too busy, but the big picture being the top box being people who had low genetic risk of high blood pressure, stratified further by those who had higher clinical cardiovascular disease risk score. And no matter what your genetic risk profile was, your clinical risk score led to a higher risk of the incident heart failure and the primary outcome here being composite of heart failure and ASCVD. What I took out of that was DNA is not destiny. A lower traditional cardiovascular risk factor profile offsets the high genetic risk for adverse cardiovascular events conferred by blood pressure polygenic risk score. At that time, I was also intrigued by the fact that this is well and good. This is talking about a trait which is not intervened on. Hypertension just incubates, keeps going on, and eventually leads to heart failure. But we treat these people. We treat them, and there should be a difference in response maybe. And that's what I kept looking at. And wonderful work. Nikki started at the same time. And it was eventually published in JAMA Cardiology, and it's one of my favorite works. Is on the left-hand side, you see these are quartiles or quintiles of the polygenic risk score of blood pressure, and they looked at populations in a clinical trial where they were giving blood pressure drugs, and the response to blood pressure drugs was different across this genetic risk score. And I see this all the time in clinic. I give the same medicine to same phenotype of 5 patients. All of them don't have the same response, even if they're 100% adherent. It made sense. They also were associated with higher odds of treatment-resistant hypertension, the exact phenotype I see in clinic. So there was a lot of this genetic background I felt was contributing to heart failure. All well and good, but we understand DNA is not destiny. And I kept thinking, but does all of this leave an imprint on the myocardium? Does it actually cause molecular changes? I didn't have evidence of that, and I wasn't trained in basic science. And now I'll tell you how I reached for my exposure into what I call translational exposomics. I started to collaborate, reach out for help, and answer questions. I worked with Dr. Holliday, who's now a basic scientist at University of Florida, and his group, and wanting to look at physiological stressors. And I'm not going to make you read through the basic science studies, but big picture was ALOX5 is a leukotriene that is responsible for leukocyte activation, inflammation after you have an acute myocardial infarction. We saw that it orchestrates inflammation after a physiological stressor event, and then also manages how you repair your myocardium afterwards. Similarly, there was sphingosine 1-phosphate. We looked at it again from mouse data, from human heart tissue data, that this was a bioactive inflammation mediator, and when it's dysregulated, leads to development of heart failure. Again, the insult being a physiological stressor that I provided. And lastly, at that time, omics was coming up and along in population space, hadn't percolated down to basic science, and we took that omics approach to basic science. And we saw that there is a different lipidome profile among those hearts which have an ischemic left ventricle, and then the hearts which go through the whole injury spectrum, from healthy to acute MI, and then eventually chronic MI, which is heart failure. What I learned from that exposure in basic science was stress exposures etch themselves into the molecular biology of sarcomeres. but these are all physiological stressors. And I really wanted to zoom into chronic psychosocial stress. I wanted to understand what you experience throughout your life. Does that have an impact on somebody's heart? Does that have a mechanistic impact on somebody's heart? This is my favorite figure out of all the works that I've done. This is a map of Birmingham, and it's overlaid with neighborhoods, and the red to blue spectrum is essentially the area deprivation index. The higher area deprivation index, being in red, are the most disadvantaged populations, with area deprivation ADI being a surrogate for the chronic psychosocial stress that one experiences. In that 7-mile radius, passing right through UAB, the life expectancy drops from 66 to from 83 to 66. That's 7 miles, 17 years. Just a little bit of a walk in either direction from here, it changes what happens to you. Your life's trajectory is already pre-decided at this point. That was really overwhelming for me. And I said, I need to dig in more deep. But I kept thinking even during that time, I wish I could study these chronic psychosocial stress mechanistically. I wasn't satisfied with this epidemiological evidence that I had. And I was looking for that deep heart to vary explanation that made sense across various levels. So I looked across the campus, tried to find people who would help me answer this. And I found these. These are cardiac organoids. Again, I find it extremely cool. These are essentially, I would take one of you all, take your blood, and make those cells into stem cells, and then divide them and culture them so that they become heart cells. And not just heart cells, they show heart cells, endothelial cells, fibroblasts, all of it. And they beat exactly like a human heart. And I felt this is where I could actually test this out. These are really cool, and I wanted to share and reach out to people, and it's like, hey, teach me how to do this. And that's what I started doing. I pivoted, and I said, I want to learn more. This is where I'm going after my conjecture stress test. I want to see if that conjecture makes sense. These organoids take a while to grow. It takes around 28 days to just grow. I was doing experiments earlier this morning trying to grow them. But they grow into neonate cardiomyocytes, or a fetal cardiac organoid, like how a fetal heart would look like. And we mature them through pressure volume loops and develop them into adult cardiac organoids. And I said, this is a great framework for me to test out my hypothesis of chronic psychosocial stress leads to inflammation, and that inflammation leads to mechanistic changes in the sarcomere and would lead to eventual heart failure. And we looked at which cytokines were produced more. I won't make you go through all of that, but the big picture was there was a robust induction of pathways of immune cell recruitment, inflammation, angiogenesis, and apoptosis. So it started to make a little bit more sense. But I still haven't answered everything, and that's where we're headed. What I learned out of this whole picture was that exposures matter at every level. At genetic level, where you're born, how you get treated for a common risk factor, and then at the cellular level. What remains is for us to quantify that, integrate that, and to be able to intervene on that. Exposomics and heart failure is big, it's large, it's unanswered, there's so many questions, and that's what I'm here to do, is to inspire you all to keep pushing it. There are a lot of unanswered questions, but they're not unanswerable. There are problems in exposomics too. If you think about it, there's problems with any scientific project that you're ever going to envision. There'll be issues in measurement, and there'll be issues in methods that you got to tackle. And we're still understanding how to do that in exposomics. We need better spatiotemporal resolution of these exposure metrics. The segregation, the redlining, the toxins you're exposed to, it changes. You all were not in Birmingham all the time. You were born maybe in different parts of Alabama or even the world, and you came over here. So your exposome changed. How does that change? We don't have a good answer for that. And we need more longitudinal assessment of this allostatic load. That clinical acumen that changes every time I see you in clinic and you're doing a little bit better or a little bit worse, that is a longitudinal profile. We don't have that yet. We need better data to be able to pursue that. And then to be able to integrate it across omics, that is the proteome, the metabolome, the epigenome, Those are really nuanced concepts that we have to try and keep trying, try and integrate it all together, that conjecture has to make sense again. Then there are still problems with methods we got to still figure out. We have to understand the causal transportability of our results across cohorts. Replication crisis is real in science. We got to acknowledge it. We got to see why it doesn't work. Why my answer works in one group of people and doesn't work in another group of people. You all here will do epidemiological projects that will make sense in one cohort, and you do the exact same analysis. Another cohort doesn't work out. So there's something which we're missing here, right? And then there's the big question of equity in multi-omics. We've focused a lot on people who are typically European ancestry, so our data is all clustered from that population. I don't know how a South Asian does, for example, or somebody from East Asia does very well. I'm from India and my wife is from China, and between the two of us, we represent half of humankind. And I don't have an answer through exposomics on how I would be able to tell whether our exposome is going to lead to heart failure. And from a clinician standpoint, I don't even know what's the threshold for me to act on this. What level should I start acting on this? What do I advise? What is my intervention? So people ask me, how do you plan to address that? And this is my near-term goal, is to create a heartbeat repository, which is essentially taking people across the spectrum. UAB is a really rich resource of people. We cater to the entire Southeast. We get people who have the most rare phenotypes to the most common phenotypes and the entire spectrum of exposomic burden. As I said, 7 miles and 17 years. And I want to put them all together. Their blood, their EHR data, their allostatic load, physiological measures, their genomic data, even the cardiac organoids, and if somebody gets a biopsy of their heart, that too. We get it together and make it available for people to be able to use, to be able to answer those questions in exposomics. My research plan is to build that hard-to-vary explanation of how our exposures shape heart failure and how we can truly modify them. We have to look for patterns, from maps to molecules. We have to acknowledge defeat and do constant error correction. That's what I did. One biomarker cannot explain it all, right? We learn. I learned how to layer social biology. I learned how to layer epidemiology and genomics all together and strengthen my explanation. And we need to collaborate. You'll see in this presentation, I collaborated with everyone that was possible on this campus. I collaborated with epidemiologists, geneticists, with population scientists, with nutrition scientists, circadian biologists, biomedical engineers, basic scientists. If there was an IST on campus, I probably reached out to them and say, hey, I want to answer my question. Can you help me answer that better? So collaboration is how we beat this, is to tackle this problem. And exposomics is not finished at the end of this talk. It's an ever-growing field, and our answers are only going to get better. And I'm going back to what I started with, the epistemology. Humans by default are universal explainers. We can explain anything that we understand. The only constraints we have is laws of physics. And the best part about laws of physics is it is computable. You could put all those laws into a machine, a Turing machine, or a computer, probably not this one because it took forever for it to load my presentation, but a computer, and it would be able to simulate. It'll be able to simulate what the world should look like. But guess what? We do that on a daily basis. We simulate. We understand how I'm going to walk, step. You walked from your classes, came here for the lecture. That was all constant simulation. And that's my message for not getting scared of AI, of large language models. We'll always be far away from artificial generative intelligence. That is creativity. And that is what you all have, is unbounded creativity. All returns in life come from compound interest. I learned that the hard way. You have to play long-term games with long-term people. And I treat heart failure as a long-term person. So here's my invitation. Let's compound our knowledge together. Especially when I see a lot of my scientific colleagues feel so disgruntled and blame every social scenario and political environment for not being able to pursue their research ideas. Let's play the long game. let's keep questioning, let's keep refining, and let's keep collaborating. Because the only revolution worth fighting for is the scientific revolution, the patient iterative discovery of better explanations. From streets to sarcomeres, let's keep growing. Thank you.